All right, we're looking at Unit 3, Lecture 6, lymphocyte response. In the previous lecture, we talked about some of the broad aspects of the third line of defense, which involves lymphocytes, and kind of described the characteristics of lymphocyte development and what they look for in terms of antigens or epitopes, to be specific. So here we're going to start off with what are the kinds of lymphocytes involved in the immune response and what are some of their characteristics. So let's start off with B lymphocytes. One of the most important types of lymphocytes overall are plasma B cells, or plasma cells in general. These are B cells that specifically secrete antibodies. Memory B cells are those that have a long duration in the body. They quote-unquote remember the antigen, and this ultimately is where the term adaptive or acquired immunity comes from. It sort of implies that you have a long-term ability to respond. And finally, regulatory B cells, not really discussed in detail here, are cells that ultimately allow the body to suppress immune responses so that we don't have too much reactivity going on when it's not needed. And these certainly represent a more nuanced part of the immune system. So we'll mention these here without going into much detail later. But ultimately, they kind of help dampen the immune response because you can certainly go too far, and they help kind of bring the immune system back down to a more moderate level. But the main two types that are important in our discussion today are plasma cells and memory B cells. Now, T cells or T lymphocytes, we have helper T cells, TH cells for short, and as the name implies, they assist in other aspects of the immune response. We'll talk more about what that means. Cytotoxic T cells, the term cytotoxic means cell killers, and what they actually do is kill the body's own cells that are compromised. And this usually implies cells that have infection by virus, or it could even be cancerous cells. But they're cells that are no longer viable, and so the best solution is to remove them or kill them. Kind of like regulatory B cells, we also have regulatory T cells, or Tregs. Similar to the regulatory B cells, they maintain a moderate response without having too much of an immune response. And similar to memory B cells, we also have memory T cells. So both memory T and B cells together are responsible for the adaptive, acquired, long-term aspect of the third line of immunity. Now, the third line of immunity can be activated in really one of two ways, one of which is the most common. The most common is called T-dependent activation. What this requires is that lymphocytes have to respond to the antigens, the foreign molecules, by having them first displayed on an antigen-presenting cell. Now, this does include a couple different types of cells, but most commonly it's a dendritic cell. So a dendritic cell is an innate immune cell, and they represent part of the non-specific immunity. But what they do is they digest and display parts of antigens on the cell in a way that then goes on to activate T cells. Usually it's the T helper cells first, and then you set off a subsequent series of reactions in which cytotoxic and or B cells are then activated downstream. A less common but still relevant mechanism is called T-independent activation. And this occurs when certain types of antigens can bypass the antigen-presenting cells and directly stimulate B cells, which then go on to produce antibodies. There's only certain types of antigens that do this, but when it happens, it's still relevant. So, as I mentioned, in most cases, you have T-dependent activation. T-independent activation occurs with certain types of large polymers, things like polysaccharides, for example, with lots of repeating subunits. Another example besides general polysaccharides is the molecule lipopolysaccharide, or LPS. So ultimately, you can activate the lymphocytes in one of two ways. Most of the time, it's T-dependent, meaning that you require an antigen-presenting cell, such as a dendritic cell, in order to present the antigen to the lymphocytes. So more about that process here. APCs, as they're often called, these are phagocytes. So recall back from previous lessons, phagocytes are cell eaters, And as we see here, the cell engulfs the antigen, digests it into little tiny parts, breaks it down, and then takes parts of the antigen and displays it on the cell surface. Then from there, it will travel to different parts of the immune system, like the lymphatic system and the lymph nodes, and display it to helper T cells, which are usually the first cell in the next step to begin that process of activating additional lymphocytes. Now, antigen-presenting cells are generally non-specific. They reside near portals of entry where pathogens will generally enter the body somewhat randomly. They have the ability to recognize pathogen-associated molecular patterns, which are PAMPs, via pattern recognition receptors, which are, again, part of the non-specific or innate immune system. Now, they recognize general foreign pathogens, break them down, but then end up displaying very specific parts of the pathogen on the surface of their cells. That then means that highly specific parts will end up being needed later to activate T cells. And I'll try to make more sense of that. But the non-specific response transitions into the specific response, in which antigen-presenting cells gobble up general foreign pathogens and then display the specific parts of the pathogen on the outside of the cell. Now, this figure makes it look like only one part of the antigen is being displayed, or one part of the microbe. But in reality, lots of parts, each one of these receptors could contain a different piece of the microbe. So the microbe may contain 100 different antigens. And in theory, an antigen-presenting cell, could display all of those different parts or a fraction of those different parts, depending on the exact context. So lots of different parts of the microbe are displayed, and that then gives the antigen-presenting cell a mechanism to activate the specific lymphocytes downstream from there. So once they have those displayed, they will migrate, as it says here, to secondary lymphoid organs, which is usually the lymph nodes. And then from there, they actually have to just bump and migrate through that organ system until they just so happen to find a matching T cell with the receptor, a helper T cell usually, with the receptor that happens to match that antigen. So that's what this figure here is trying to show. This is the antigen presenting cell. This represents a dendritic cell. This represents some of the foreign processed antigen here. And then this represents the interaction with the T helper cell. Down at the microscopic level, the T helper cell has to then have a specific receptor that is capable of binding that specific antigen in the shape of that antigen. So if you recall back to the previous lesson, in the first part of chapter 16, we described how VDJ recombination gives rise to billions of different lymphocyte receptors. So in your body, you have billions of different variations of these T cell receptors. And one of those, just by chance, will bind the multiple antigen types displayed by the dendritic cell. Since the dendritic cell is going to display potentially hundreds of antigens, it becomes much more likely that at least one of those lymphocyte T cells will have a receptor that matches. But this takes some time, and it gives an explanation for why the third line of immunity is not activated immediately. But once that T cell with a matching receptor is found, it will now become activated, and it will now lead to the next stage, which is the activation of the helper T cell. So the antigen presenting cell has to activate that helper T cell. Before it's active, it's referred to as a naive T cell, meaning it has yet to be encountered with an antigen. So the antigen presenting cell migrates, finds the antigen finds the T cell with the matching antigen. Going back to the major histocompatibility complexes, antigen presenting cells can use both types 1 and type 2. Helper T cells specifically will recognize the antigens on class 2 MHC complexes, whereas cytotoxic T cells will recognize antigens presented on class 1 MHC complexes. Ultimately, the reason why that matters is that helper T cells will go on to activate B cells, which release antibodies, which tend to circulate and do their damage outside of cells. Cytotoxic T cells will bind antigens presented from the inside of cells, and their job is to remove things like virally infected cells or cancer cells. And that is a manifestation of a problem happening inside of the cell. So each of those receptors have a specific goal in mind, which is either to activate things that will clear up problems on the outside of the cells, which would be the helper T cells, or to remove cells that have internal problems, which is where cytotoxic T cells, they remove the problem by killing the entire cell itself. Now, once the antigen presenting cell migrates, it is going to set off clonal selection and expansion. We did talk about this last time, so just to kind of reiterate that here real quickly, that the matching antigen is brought to the receptor in which we have a match, that is, and that is now said to be selected. And since a pathogen has a multitude of antigens, each antigen with multiple epitopes, this increases the likelihood that you'll find a match somewhere in the lymphatic system. So a single pathogen can activate a wide variety of specific lymphocyte clones, providing the body with lots of different ways to activate cells. So it seems unlikely that you would find a matching lymphocyte that just so happens to have the right receptor until you realize that actually you could be activating any one of several hundred different types of lymphocytes that all happen to respond to different parts of the antigen. So it actually is much more likely than it might seem initially. Once that particular version of the T cell is activated through selection, clonal expansion of the helper T cell in this context now takes place. So now you'll have lots and lots and lots of copies of just that one helper T cell. And those helper T cells now have to go on and find a B cell with an identical matching receptor, or one that's close enough. And so now what we have to do is wait even longer so that that one set of cloned helper T cells goes out and finds at least one B cell, a naive B cell, that can now have a similar identical matching receptor. This takes even more time. You have more migration that's happening. Once a matching B cell is found, a naive B cell, the helper T cell will now activate that B cell and stimulate the naive B cell to turn into a plasma B cell or a plasma cell, which will then go on to start secreting antibodies. And it's really then the antibodies that start to have the effect, not necessarily the B cells themselves. This is a complex process, and it's worth noting that as we'll talk about at the end here, but just to kind of preview this, the adaptive line of the immune system, it may take days, if not even a week or so, to activate. It takes a lot of time. It is somewhat variable, but this does not happen overnight. It usually takes at least several days to get it going, if not maybe a week or 2 weeks. So a lot of that time delay is explained by the process by which the antigen presenting cell has to take the antigen to the T helper cell, called the CD4 cell. That cell now clones itself, making copies of different variations of different helper T cells initially. And then the helper T cells now migrate even further, finding a specific matching copy of a B cell. And that B cell now goes on to clone itself into multiple variations of B cells, one of those being a plasma cell, which then goes on to produce the antibodies. It takes time for all that to happen. Now, once the B cell is activated, it will start to differentiate into the short-term, what we'll call effector cells. I'll describe those two terms, effector and memory cells, at the very end. But effector cells are things like plasma cells that have an immediate effect against the pathogen. In other words, they go right into action. Memory cells don't necessarily activate immediately, but hang back for long-term immunity. And this is what gives us the long-term acquired immunity. Antibodies go on to have an effect, as I mentioned. We'll discuss what they do specifically here shortly. Antibodies are kind of like a secreted cell receptor. So we've talked about how the cell receptors are generated using VDJ recombination and how receptors have lock and key specificity. Receptors are the mechanism by which cells kind of feel out what is foreign and also what is self versus non-self. So what you need to recognize is that antibodies are essentially just a receptor, except they don't remain attached to the cell in most cases. They're secreted in high volume, so that the same concept of lock and key specificity can take place outside of the cell. So they do their work binding to antigen external from the B cell, and ultimately they can flood the body and saturate different cavities of the body in ways that the individual cells themselves cannot. This is different from T cells. T cells have similar kinds of receptors, but their receptors remain attached to the cells at all times. And there are advantages to that. There's advantages and disadvantages, I should say. But it's just important to note that specifically it's the antibodies that are like a quote-unquote secreted cell receptor. They function very much like a receptor, just with being external from the B cell. One of the main advantages there is that a cell surface can only hold so many antibodies. Once they're secreted, you can produce far more than what you could hold on the surface of the cell. So it gives the body a greater advantage in that more and more of these receptors can be pumped into the body, whereas T cell receptors can't do that. Ultimately, antibodies play a role in clearing things that are external to the cells. So viruses, by contrast, can stay inside the cell. They can be both inside and outside. But once a virus leaves an infected host cell and it migrates outside of it, antibodies can now start to bind to the virus and neutralize the virus in that particular context, outside of a cell. However, if a virus is still inside of a cell, such as they are when they're replicating and using the host cell for replication and protein synthesis, for example, in that location, antibodies can't really attack them, and that is where you need things like cytotoxic T cells. So antibodies are good for clearing things up in the extracellular environment, where cytotoxic T cells are essential for clearing out the cells themselves that are infected so that the virus can't continue to replicate. And I'll just elaborate on that a little bit more and then I'll move on. If the virus was only cleared in the extracellular environment, then in theory, which is what antibodies do, then in theory, the virus could continue to replicate and continue to spread. So by neutralizing the virus outside the cells, that keeps the virus from infecting new cells, but doesn't address the cells that are already infected. So the cytotoxic T cells then remove the virally infected cells so that they can't keep replicating, and then the antibodies neutralize all the viruses as they attempt to escape one cell and then infect another cell. So it really, you kind of have a 1-2 punch, so to speak. Cytotoxic T cells clear the virally infected cells themselves so that you can't keep replicating as a virus. Antigens then neutralize the virus particles as they try to move from one cell to the next. And so by doing that, you ultimately clear the body from both the free-floating viruses and the cells that are virally infected. Now this figure here I'll just put up and I'll move on. This just represents what's happening here. This is a figure out of your textbook here, and you can look at this for more detail. But this represents an example of both T-dependent and T-independent activation. This arrow here represents the independent activation, some antigens directly activating a B cell. This part over here represents T-independent activation with a T helper cell. Either way, you get an activated B cell that differentiate into plasma, memory, and regulatory B cells. Plasma cells go on to secrete antibodies. Memory cells go on to form long-term immunity. Regulatory B cells play a more complex role in mitigating or sort of regulating and keeping the immune response in check. So a little bit more about the cytotoxic T cells. Cytotoxic means cell killer. And as I've already mentioned, they're responsible for removing the quote-unquote compromised host cells. When it comes to infection, this primarily involves viral pathogens, viruses that are actively replicating within infected cells. It is worth noting that some bacteria are intracellular as well. And I don't go into every little example, but mostly that implies viruses in terms of infectious pathogens, but there are some bacteria that this would apply to as well. So viruses have a little safe haven inside the cell. Cytotoxic T cells remove that safe haven. The cell has to be sacrificed, but it ultimately is a sacrifice that is a net benefit to the overall body. Cytotoxic T cells have to be able to recognize and destroy any virally infected host cell, and that is done through the MHC complex proteins, MHC1, MHC class 1 proteins. And as they're sampling antigens from the internal contents, viral antigens will end up being displayed on infected host cells. And it's a signaling alarm that tells the body that these cells are compromised. Once that recognition takes place, the cytotoxic T cell destroys the cell with a mechanism that involves destructive enzymes. And those include enzymes called perforins that puncture holes in the cell, and granzymes that break down critical proteins necessary for cellular function. So that's kind of the specific mechanism without going into the detail here. There are specific kinds of toxins and enzymes that are released that actually do the damage to the cytotoxic, or so that from the cytotoxic T cell that break down the host cells themselves. This is why, by the way, regulatory cells, like regulatory T cells in this case, are so important, because if you allow that process to go on for too long, you can just damage your own body just as much, if not more, than what the virus itself can. So that process is very much a double-edged sword. Now, going on here to the antibodies, we'll kind of finish up this discussion largely focused on antibodies. So as I mentioned, antibodies are like secreted cell receptors. These are more technically known as immunoglobulins, or Igs for short. A globulin is a sort of a large protein, so that word kind of means large immune-based proteins, something like that. Antibodies are variable, or they have different variations, I should say, but they have two key regions. You have what's called the variable region, and these are the antigen binding sites. And what antibodies have is at least two different antigen binding sites. It's almost like you having two arms. Like if this was your body, you have two arms, and each arm can each bind an antigen, and in this context, each antibody can bind two antigens on each separate arm. So the arms are effectively the variable region, variable in that they have the capacity to bind different types of antigens. The constant region, sometimes known as the FC region, this region is constant in that it doesn't really change much from one antibody to the next, and it is used to bind to the host cells like macrophages, for example. So variable region, two of them have the specificity. Both arms can bind the same antigen. And when we get to the functions of antibodies here in a minute, that will make a little more sense as to why that's necessary. I'll try to reiterate that. The constant region is constant because it doesn't really change. Constant is like a non-changing variable. It's the same. And ultimately, this allows what you might call the stem portion of the antibody to be used as a mechanism for your white blood cells to grab a hold of it. So this part can grab a hold of an antigen, and this part here can then cling to your white blood cells. And that actually becomes a mechanism for some of the functions that we'll see here in a second. Antibodies don't always have a perfect fit the more close the fit is, the more stronger the activation, like you see here. Sometimes you have, so you either have like a nice perfect fit or no fit, or there is sort of in between, which is a poor fit or weak activation. And that's where you get a lot of nuance in the body. It's not everything has to be a perfect fit. There is some wiggle room, and that can kind of determine the strength of activation. Now, once antibodies are created, they will generally do one of four things. Opsonization is a term that was mentioned in chapter 15. What this implies is that the antibodies will bind to the pathogen with the variable region, and then the FC constant region sticks out and then is able to be grabbed by phagocytes, which allow them to be gobbled up into the white blood cell. So that process is sort of a mechanism by which the antibodies can be used to bind to the pathogen and then act as a mechanism for the body to pull the pathogen out into digestion. And here's a figure of what that looks like here. So this is the variable region of the antibody binding to the pathogen, and then the constant region sticks out, and then the things like macrophages will grab the constant region and use that almost like a handle to bring it into the internal part of the phagocyte, the macrophage more commonly, and basically it's an enhanced mechanism of phagocytosis. Once something like a bacteria is opsonized, it's much easier for the bacteria to be phagocytized by the immune system. Agglutination is where multiple arms of the antibodies are necessary. So one arm, as you see here, can join or bind to one bacterium, and the other arm can bind to another bacterium. And that way, a single antibody can cross-link or agglutinate two cells together. And then once that starts happening in a large complex, what happens is lots of bacteria can all be kind of contained by a small number of antibodies, kind of like you see here. That's the idea of agglutination. It keeps bacteria from spreading. This is especially important for mobile bacteria that can spread throughout the body. That keeps them from spreading. Neutralization works largely with things like viruses, but also things like toxins. Neutralization, when you look at how a virus works, think back to the chapter 7 on viruses. The virus has something like a spike, and that's the kind of the key of the virus that has to be used to infect your cells. Antibodies essentially cover up the spike, and it prevents the virus from being able to attach to the host cell. So as long as the virus is covered up in antibodies, it can't go on to infect new cells. That also applies to toxins. So toxins are little molecules that have to bind to some kind of cell typically before they cause an effect. So if the antibodies bind to them first, they neutralize the toxin. And the last thing here is lysis. This is part of the complement system. We talked about complement in the last chapter, chapter 15. One of the mechanisms of the complement system is that the complement proteins bind along with antibodies, and the complex between complement proteins and antibodies end up puncturing holes in the membranes of things like bacteria primarily. So that's sort of a simplified bullet point on a more complex process, but that's what that means. Lysis is part of the complement system, and the antibodies play a role in puncturing holes. So those are somewhat broad explanations for each of those, but that's essentially what your antibodies end up doing. And just to reiterate something I said earlier, all of these processes take place in the extracellular environment. Anything going on inside the cell can't really be affected by an antibody. So these are all things that take place in the extracellular environment. Now, there are about 5 broad classes of antibodies, and a lot of these can be broken down into more specific subclasses, but these are the 5 broad categories. So you want to be familiar with the 5 broad types and some of the basic functions here of each. You can see the study guide for the specific questions that I will ask. IgG, immunoglobulin G, this is the most abundant out of all the antibodies. It's also the one that can cross the placenta. Tends to be the most effective overall for things like opsonization, neutralizing, activating complement, but also just more specifically when you look at how natural passive immunity works, this is the one that provides natural passive immunity, which I'm having you read out of the textbook. Natural passive immunity is how a mother provides immunity to a baby in utero. So the baby can't necessarily use all of the antibodies from the mother, but it can use the IgG antibodies, which are the most abundant. IgM is the first one produced by plasma B cells, and it also has a more complex shape. It ends up having what's called a pentamer shape. And I'm just going to go back here real quickly to a previous figure to kind of give you an idea of where you can review this here. So this figure here shows the activation of B cells. Plasma cells are activated, and then what you see them producing here, these little pentamer shaped, you see the little Y-shaped antibody like I showed you there. There's actually 1, 2, 3, 4, 5 of those. That's a pentamer shape. That's what that is. So the pentamer is a representation of the IgM antibody complex. And that is a specific form that is produced first in the process. And that ultimately then gives the pentamer shape gives it more arms. Instead of having 2 arms, every pentamer shaped IgM actually has 10 arms. So it makes it superior for agglutination. IgA is one that, kind of like the IgM, it can be joined together, but this one can be joined together as a dimer. The 2 constant regions of an antibody can be joined, and these can then have 4 arms instead of 2. But the main advantage here is that IgA is key for mucosal immunity. Mucosa is part of the innate barriers to defense, and over time, the IgA antibodies become infused in the mucosa, and so they play a key role in helping strengthen the mucosal defenses, which are part of the innate immune system. IgE plays a role in allergic reactions. So anytime you have an allergy, especially allergies that relate to histamine, IgE is a key part of that. We'll talk about that in the last chapter of the semester. That's chapter 17. We'll talk about the allergic, how allergies work, and the what's called a hypersensitivity reaction. And then IgD, the only thing you want to know about this one is that some of the antibodies do remain attached to the B cell. So I mentioned that antibodies are like secreted receptors. The one exception to that is IgD. IgD is the only one that actually stays attached to the surface of the B cell. Okay, a couple more things here and we'll wrap this up. Antibodies are not produced in a linear fashion. They are produced in sort of a staggered type of response. So upon first encounter with an antigen, this is called the primary response, what you have is a long delayed process involving the antigen presentation, activation of helper T cell, and then subsequent activation of the B cell, division or differentiation into a plasma B cell, and then the secretion of the antibodies. That whole process takes time, and although the process is unfolding, to you, nothing is really happening. As you're getting sick, your body has not actually started defending itself yet in the context of the third line. So this whole process involves what we call a latent period. And in that time period, it doesn't seem like anything's happening because you haven't yet really started to fight the pathogen. Now, another key aspect of the primary response, other than that it involves a latent period, is that IgM is the main antibody produced. Now, in a secondary, or what is called the anamnestic response, this could be really the second or any subsequent response after the first. At the end of the primary response, you get memory cells. If a memory cell encounters a pathogen later on, it can immediately differentiate into a plasma cell. Thus, there's no latent period. So a secondary response has no latent period, and therefore antibody production occurs immediately. Also, memory cells are different in the way they respond than the naive B cells that are initially activated. And so there's additional features that we'll get to here in a second, comparing the two types of response. But the big difference between a primary and a secondary response is that there's no latent period because the memory cells are activated directly and don't require antigen presentation. So on a graph, it looks kind of like this. First exposure occurs here in time, latent period here, and then we get antibody production here. You can see from the two colored lines how IgM and IgG responses play out, kind of the total antibody production here, and then antibody production comes back down to baseline. However much time occurs between the first and secondary exposure, the secondary exposure starts here and immediately antibody production ensues. Not only is there no latent period, but the amount of antibody production goes up above the amount in the first exposure. And as we see here, more IgG, less IgM. Both are produced in each response, but a different amount of each one. So that ultimately is what we mean by no latent period and a different type of response between the naive B cell and the memory B cell. So to summarize that, the big difference is a latent period in the primary response, which is absent in the secondary response. All these aspects are complicated, so there's lots of variation in how long a latent period may be, but you can kind of think about it in your own experience. You start to feel symptoms. This is what we call the prodromal stage of infection. You sore throat, headache, kind of feel tired. Your body is now starting to sense and respond to the pathogen. However long it takes you to really clear it out kind of gives you an idea of how long that latent period exists. Not the most perfect way to measure that, but kind of anecdotally you can kind of get a sense for that. Maybe a couple days, right? But in reality, it actually takes longer to fully clear it. But you might be back up and running again within a day or two. That kind of gives you an idea about how long it takes. But in reality, it actually takes a lot longer. Your body isn't aware of every little detail here. Another key response of another key part of the secondary response is you get greater amounts of antibody. This has to do with the way memory cells are quote unquote programmed to produce greater amounts of antibody. Antibodies then persist longer in the body. This is because memory cells will produce them for a longer period of time. So not only do you get more antibody in a shorter period of time, but it lasts longer. And this will increase with each different subsequent response. So one of the reasons why they sometimes call it an anamnestic response is it sort of implies after the first. And if you get exposed a third time, you'll have even more antibody than the second time, and it will last longer than the second time. And if you get exposed a fourth time, the numbers keep going up. And this is kind of the logic behind booster shots, theoretically, that the more, if you're talking about vaccinations, good vaccinations that actually work, then what you're talking about is that the vaccines are designed to sort of simulate a secondary response, or booster shots, that is, are designed to simulate a secondary response, or maybe even a third or fourth, or however many booster shots you may need. Maybe you need 100 of them, who knows. But those booster shots are sort of like artificial exposures to pathogens, as if you might have been exposed to the real thing. And each booster shot theoretically kind of boosts that immunity up higher and higher, again, assuming that you have vaccines that actually work. To summarize the immune response here, the third line is characterized by sort of two general types of cells. The effector cells are those that have the immediate action, plasma B cells producing antibodies, cytotoxic T cells killing the infected cells, helper T cells sort of mediating the response. Long-term immunity then is conferred by the memory cells, memory B cells, and memory T cells that each play more nuanced roles. Memory B cells tend to circulate throughout the body, whereas memory T cells tend to stay in the tissue where they were initially activated. Everything that we've talked about here has layers upon layers of complexity. These are fairly detailed notes, but nowhere near as complex as the full detail. Memory T cells are, in particular, quite specific. Much more detail there. But anyway, that kind of gives you a pretty good idea of how it works. This little figure over here, just to point out, shows a naive B cell being activated into a plasma, and then a memory cell. Plasma cell goes on to produce antibodies. Memory cells can be activated, differentiating into a plasma cell that then goes on to produce antibodies. So that's a similar thing happens with T cells, but not quite as concisely there. So anyway, that ultimately kind of gives you a nice summary of the immune system. Once they're activated, go into effect, hang back for long-term immunity, and that's ultimately where the term acquired or adaptive immunity comes from. All right, we'll wrap it up there. Make sure that you look over the textbook for the different types of immune responses, natural versus artificial, and active versus passive. And I'll have some examples, or I'll have some questions in the study guide rather, to make sure that you've looked over that and some questions on the quiz and exam. All right, see you later.